Hey y'all, my name is Sammy the Salmonella. And my name is Sid the Streptococcus. Today's lesson, we will be going over horizontal gene transfer in prokaryotic cells. But before we get started, shout out to Doc Martin at the University of Puget Sound for this lesson that he requested. Let's get right into it. HGT, also known as horizontal gene transfer, is a way for bacteria to achieve genetic diversity. It is also an important factor in prokaryote evolution. But on the downside, it is a main way to build up antibiotic resistance in bacteria. There are three mechanisms when it comes to HGT in bacteria. The first one is conjugation, which is also the most common. Then we have transduction, and finally, transformation. Like I mentioned before, conjugation is the most common mechanism in horizontal gene transfer between bacteria. There is a donor and recipient cell. The donor uses a pilus, which is an appendage that attaches to the recipient bacteria. And from there, the DNA is transferred between the two cells in the form of plasmids. Plasmids are small circular DNA molecules found in bacteria and some other small microscopic organisms. What is cool about this is that the donor cells have a chunk of DNA that is called the F factor, which stands for fertility factor. Once this is transmitted to the recipient, it is able to make its own pilus and transfer DNA to other bacteria cells. A random but fun example of this is when you're in a zombie apocalypse and you get bitten. You end up turning into a zombie, which is kind of exactly what happens with bacteria in the sense of using this conjugation mechanism of HGT. Next is transduction. This happens when DNA is transferred from one cell to another via bacteriophages. Bacteriophages are a type of virus that infect bacteria. There are two types of transduction, generalized and specialized. Generalized transduction occurs when a bacteriophage can pick up any section of its previous host genome and inject it into another bacteria. On the other hand, specialized transduction happens when the bacteriophage can only pick up a specific portion of the host genome and inject it into another bacterial cell. Once the bacteriophage infects another host cell, it turns them into viruses to carry on and infect other bacterial host cells. How crazy is that? Last but not least, transformation occurs when bacteria absorb free-floating DNA from their environment. They also might come in the form of plasmids. The bacterium that interacts with the plasmid takes it in and passes it on to other bacteria cells that come from that bacterium. This could be a problem because the plasmid that it takes up could turn a harmless bacteria into a pathogenic one, and we don't want that. The main takeaways from this lesson are that HGT is important for prokaryotic evolution. It shares DNA that can have no effects or beneficial genes that are passed down to descendants of that bacterium, and it creates a larger diversity that is facilitated by natural selection. On the other hand, it could spread parasitic genes across bacteria, and it could result in building stronger antibiotic resistance. That's all for this video. I just want to thank you for watching and have a great rest of your day. Bye!